Hello all, hope you all are doing well. So let's continue with our topic that we started in the last class, transportation. So if you remember in your last video you studied about structure of heart. Now the next part of that we have here is about what is blood and its components. So simple question, what is blood? So blood is nothing but a type of a tissue, connective tissue. You already studied types of tissues in your lower class. So blood belongs to connective tissue and it is the only connective tissue which is in liquid state. So blood is a liquid or a fluid or a vascular connective tissue and it is most abundant connective tissue in our body. Now when you talk about blood, you should know that study of this blood is known as hematology. Study of blood and its components togetherly referred as hematology. Now what this blood is made up of? So this blood is made up of two main components. If you take this blood in a test tube and allow it to centrifuge for some time, settle down for some time without letting it clot, then that blood gets divided into two main components, and those two main components are the solid part and the liquid part. The solid part that settles down is known as blood corpuscles, and the part that floats is known as a plasma. Okay, so blood corpuscles and the plasma are the two main components of the blood. This plasma is about 55% of the total blood and remaining 45% is blood corpuscles. Now what is there in this plasma? When we study detailed analysis of this plasma, we can come to know that it consists of about 90% of water, then 7% proteins, which can be called as a plasma proteins, very important part of the blood. And that plasma proteins contains various kinds of proteins, like when blood comes in contact with air, it clots. So this clotting is because of one protein called as fibrinogen. Similarly, there is albumin, then globulins. So globulins are nothing but antibodies which are very important for fighting against diseases. So they are the proteins, 7% of the total plasma, and remaining 3% are the other substances. Now what comes in other substances? So that is nothing but gases, respiratory gases in the form of oxygen, carbon dioxide. There are wastes, the nitrogenous wastes like ammonia, or there is urea, uric acid. Then there are nutrients which are absorbed in the blood in the form of glucose, fatty acids, etc. So those are other substances like gases, antibodies, wastes, especially nitrogenous wastes, etc. Now what comes in the blood corpuscles? So when we say blood corpuscles or blood cells, simply, it consists of three main blood cells, and those three main blood cells we can call them as RBCs, red blood corpuscles, WBCs, white blood corpuscles, and platelets. So scientifically they can also be called as erythrocytes. Remember the name. RBCs or erythrocytes. WBCs or leukocytes. And platelets or thrombocytes. Got it? RBCs or erythrocytes. WBCs or leukocytes. Platelets or thrombocytes. These blood cells or blood corpuscles are about 45% of the total blood. So when it comes to what are the functions of the blood, these are the functions of the blood, like it has different proteins which help in clotting of the blood, provides immunity, then it helps in transport of gases, especially that is also the function of RBCs. It helps in transport of the gases, transport of wastes containing nitrogenous wastes like ammonia, urea. Then when it comes to RBCs, the main function of RBCs is they help in transport of gases like O2 and CO2. WBCs are nothing but the soldier cells in our blood which fight against pathogens, disease causing agents. Platelets or thrombocytes are basically concerned with clotting of the blood whenever we get an injury, any cut and a blood flows. So that should not continuously flow. So to avoid that loss of the blood in the form of bleeding, we have got these platelets or thrombocytes. Alright, so I hope you got an idea a little bit about what is blood and what it is made up of. Now we are going to deal in little detail about these types of blood cells one by one. So we are going to begin with the first one, RBCs. Okay, so it's time now to start with the first one, RBCs. Technically, it is called as erythrocytes, right? So RBCs or red blood corpuscles. These are the most abundant blood cells compared to these three types: RBCs, WBCs and platelets. So these RBCs are produced in bone marrow cavity. In fact, all blood cells are produced here only. Bone marrow cavity. Then also there are some more organs where these are produced, like it is also produced in the liver but not in adults, only in infants. Then it is also produced by the help of one hormone produced by kidney also, like erythropoietin. Now these many details you don't need. Just remember this major organ where RBCs are produced. Even spleen, another organ located in the abdominal region just close to our stomach, which is also called as a graveyard of these RBCs, because it produces RBCs as well as it destroys RBCs. Now these have got a definite shape. We all know they look dumbbell shaped, biconcave. So RBCs in mammals as well as birds, now they look like dumbbell shaped. They are biconcave, very small, about 7 micrometer in diameter, and it contains, now if you see the shape, it will look like this, dumbbell shape. In front view it will look like this, oval or elliptical, and it has got something inside responsible for transport of gases, the pigment hemoglobin. So in these RBCs, they lack other cell organelles, like nucleus is absent when they are mature. In order to occupy more and more place for hemoglobin, there is no nucleus. But in camel, there is nucleus. Remember that exception. Now they have got a lifespan of about 120 days. The lifespan of these RBCs is about 120 days. And now comes its main function. So the role performed by these RBCs is basically transport of gases. Which gases? So mainly oxygen and carbon dioxide. Basically oxygen. So when oxygen combines with this hemoglobin, it forms oxyhemoglobin. Oxyhemoglobin, which is then taken to the tissues. When it reaches to the tissues, then reverse reaction of the same will occur, and this hemoglobin will be separated from oxygen, and that it will be transported. Same way, when this carbon dioxide combines with this hemoglobin, it forms carboxyhemoglobin. Carboxyhemoglobin. So this is the main function of the same. Sometimes number of these RBCs increases or decreases than normal. So in that case, it will be abnormality, it will be disorder, which could be called as, like you may be knowing, anemia. So anemia is a condition in which number of RBCs decreases than the normal number. Sometimes number of RBCs increases also, like there are cases, for example, people who live in hilly belt area or at altitudes, they have more RBCs than the people who live in the seashore region. Why so? The reason is, as altitude increases, oxygen decreases. So in order to consume more and more oxygen, they need more and more hemoglobin or more and more RBCs, and this is the reason why people living in the hilly belt area or people living at a higher altitude, they have more RBCs compared to the people living in the lower altitude. Fine. So like this, there are so many diseases, like this anemia is there. If there is increase in the number, the disorder can be also called as polycythemia. Now these are not required for your NCERT as such. So I hope you got an idea what are RBCs, where they are formed, how they look, what is their lifespan, and what is the main role of these RBCs. Comfortable everybody? Just have a glance. Okay, so now it's time to learn WBCs. White blood corpuscles. Technically we call them as leukocytes, and these are the soldier cells in our body, right? They help us to protect from the infection or diseases. So comparatively, these WBCs are less than the RBCs. When it comes to the number, RBCs are about 4,000 to 5,000 million per cubic millimeter. Remember the number, 4,000 to 5,000 per cubic millimeter. But when it comes to these WBCs, these are only 4,000 to 9,000 per cubic millimeter. So number is very less, and this is the reason why our blood looks red in color, because of RBCs. Now, if you see the ratio between RBCs is to WBCs, it will be 600:1. Fine. Now where they are produced? So these are also formed in the same organ, which is called as bone marrow cavity. So they are also producing the same organ, that is bone marrow cavity. Even they are produced inside lymph, or they are found inside lymph, another colorless fluid which we have already discussed in the previous video. So these WBCs also have a definite shape, you can say. Various shapes they exist in. So depending on their shapes, they can be classified into 5 types also. So they show different shapes. So we can say polymorphic nature. Polymorphic means they have different external features or morphology. Size varies again. There are different types. So size may vary from like 3 micrometer is the smallest one and may go up to 100 micrometer also. Now when we talk about types of these WBCs, so they can be broadly divided into 2 main types, and those 2 main types are granulocytes and agranulocytes. So there are 2 main types of these WBCs, granulocytes and agranulocytes. What is the meaning? Granulocytes are those WBCs which have granules in their cytoplasm. And agranulocytes, they do not have granules in their cytoplasm. So these granulocytes are of 3 types. That is, first type, eosinophils. Even they can be called as acidophils, basophils, and neutrophils. See, there is a logic behind these names. Acidophils or eosinophils, basophils, neutrophils. Why they are called so? When we observe these WBCs under microscope, we need to use stain, dye. So if you want to observe eosinophils, you will need eosine, which is acidic dye. Basophils, they can be observed by basic dyes like methylene blue. Neutrophils require neutral dyes to be observed under microscope. So that is the logic why they are called as acidophils, basophils, and neutrophils. When you talk about agranulocytes, they are comparatively less than granulocytes, and there are 2 main types of this agranulocytes like monocytes and lymphocytes. So these are the 2 types, monocytes and lymphocytes of agranulocytes. Now when you look at this shape of their nucleus, basically that is different in all these 5 types. So here in acidophils, the shape of nucleus is simple like this way, bilobed. In basophils, the nucleus is S-shaped. The shape of nucleus is like S. Neutrophils have multi-lobed nucleus. So there are many lobes. So that is the characteristic feature. And here is a cytoplasm that contains granules. So they are granulocytes. Now, monocytes. These are the largest WBCs. And they have kidney-shaped nucleus. So you can remember that. M and K. Monocytes with kidney-shaped nucleus. Monkey word you can remember, which has both alphabets. And these lymphocytes are the smallest WBCs with spherical nucleus. So I hope you understood the difference. So these WBCs can change their shape, they can squeeze themselves, they can pass through the blood vessels even with the less diameter than their own diameter. So that property is possessed by these WBCs but not by RBCs. Now coming to the percentage. So percentage wise when you talk, There is one word you can remember. One mnemonic I'll give you. And that would be this. Nobody likes my educational background. Now, if you look at the first alphabet, from each word, you can see this N stands for neutrophils, which is maximum about 60 to 65%. Then there is L for lymphocytes. So it is approximately 20 to 25%. Then M, that is monocytes, which are about approximately 9%. Eosinophils, these are about 2 to 3%. And these basophils, that is background, are the least one, which are only 0 to 1%. So least WBCs are these basophils and maximum WBCs are these neutrophils. Now, when you talk about functions, basically these neutrophils, they help in phagocytosis. I hope you remember this word. Phagocytosis, which means swallowing or engulfing foreign bodies, especially germs or pathogens. Then after this, same is the function of these monocytes. These also help in phagocytosis. Likewise, lymphocytes, when we talk, they produce antibodies, which are the special chemicals which fight against pathogens or disease causing agents. Eosinophils, they help to destroy allergic substances. So this helps in allergic response. And these basophils, they produce some chemical substances, especially again as a response of this allergy. That chemical substance can be called as histamine. So these are the functions of WBCs. And the lifespan of these WBCs varies from 3 days to 10 days. When they are in the lymph, their lifespan is more. When they are in the blood, lifespan is less. Also, there are disorders related with these WBCs, like RBCs help in transport of gases. Functions of these WBCs, as we already studied, basically they help in providing us resistance against diseases or immunity against diseases. So if they will increase in number or decrease in number, diseases will be like, you must have heard the names of the diseases: blood cancer, leukemia. So this is actually the condition where number of WBCs increase abnormally. Normal range we know about 4,000 to 9,000 per cubic millimeter. But in this case, number of WBCs may cross 2.5 lakhs per cubic millimeter. If they will decrease, then it will cause low immunity, poor immunity, and that will be an invitation to pathogens, disease causing agents. So I'm sure that you have understood what are RBCs, what are WBCs, and now it's time to learn the third one, platelets. Now, the third one and the last one type of the blood cell, platelets, or we can also call them as thrombocytes. So platelets or thrombocytes are the smallest blood cells, and the main function of these blood cells is they help in clotting of the blood. So these platelets, are produced from some special cells which are called as megakaryocytes inside bone marrow cavity only. So these are the mother cells of these platelets, you can remember, megakaryocytes. Number of these platelets is approximately 2.5 lakhs. Unit is same per cubic millimeter. And as I said, they are the smallest blood cells. Smallest blood cells which look like spindle shaped. Spindle shaped means like pointed at ends and broad in the middle. Spindle shape. Lifespan is again same like WBCs, approximately 7 days to 10 days. And finally, their function. So function of these platelets is, already I told you, they help in clotting of the blood. In medical language it will be called as a coagulation of the blood. So they help in coagulation of the blood or a clotting of the blood. Blood clotting. So you must have heard about dengue patients. So in those patients, number of these platelets decreases, and that causes internal injuries, internal bleeding. That is one of the sign or a symptom of this dengue. So internal injury, internal bleeding could be the symptom of this decreased number of platelets. So there is actually a chemistry of clotting of this blood, which is little extra what I am telling you people. So it will be like whenever we get any injury, these platelets gets damaged. When they get damaged, they release one chemical substance called as a thromboplastin. Now this thromboplastin is inactive and converted into thrombopoietin. Now this is not there in your syllabus, this is for your competitive exams. Now this thrombopoietin helps in conversion of prothrombin into thrombin. And now this thrombin converts fibrinogen, which I told you, type of a protein present in the blood, into fibrin, and the moment this fibrinogen is converted into fibrin, blood clots. So this is what the chemistry of blood clotting. Platelets first release this thromboplastin. Thromboplastin gets converted into thrombopoietin. Thrombopoietin helps in converting prothrombin into thrombin, and then this thrombin helps in converting fibrinogen into fibrin, and the moment this fibrin is formed, blood clots. So this bleeding time varies person to person. In some people this bleeding time may be less, in some people it might take more time. So there is a small test how this bleeding time is checked, and the test is simply a capillary, glass capillary is immersed into a blood, and then it is shaken till that blood moving stops, setting a time. So whatever time is required for stopping that movement while shaking that capillary, we will call it as a bleeding time of that person or that individual. So I am sure now you have understood all blood cells, RBCs, WBCs, capillaries. Now after this we have got little bit to study about that lymphatic system, which is the last part of this human circulatory system, and then in the transportation we have the last part left, which is transport in plants. which will be our next video. So we'll stop here in today's session. Stay home, stay safe, take care, have a good day.